Welcome to the Scientific Sense podcast, where we explore emerging ideas from science, policy, economics, and technology. My name is Gil Eapen. We talk with world's leading academics and experts about their recent research or general areas of topical interest. Scientific Sense is an unstructured conversation with no agenda or preparation. We cover a wide variety of domains where new discoveries are made and new technologies are developed on a daily basis. We are most interested in how new ideas affect society and help educate the world how to pursue a rewarding and enjoyable life rooted in science, logic, and information. We seek knowledge without boundaries or constraints and provide unedited content of conversations with researchers and leaders who love what they do. A companion blog to this podcast can be found at scientificsense.com, and this podcast is available on over a dozen platforms and directly at scientificsense.net. If you have suggestions for topics, guests, and other ideas, please send them to info@scientificsense.com, and I can be reached at gil@eapen.info. My guest today is Professor Mark Baer, who's Professor of Neuroscience in the Picower Institute of Learning and Memory and the Department of Brain and Cognitive Sciences at Massachusetts Institute of Technology. His laboratory is interested in how the brain is modified by experience. He uses a variety of methods to examine the synaptic modifications that form the neurobiological basis of learning and memory. Welcome, Mark.
Thank you very much. It's really a pleasure to be here.
Yeah, thanks for doing this. So I want to discuss a few of your interest areas as part of your lab at MIT. And one of them is this disease state called amblyopia. Is that the right way to pronounce it?
Yes.
Amblyopia is a prevalent form of visual impairment. You say it's a disability that arises during infancy and early childhood when inputs to the visual cortex from the two eyes are poorly balanced. So it's a bit like you don't really have the early reinforcement, it sounds to me, Mark. Is that the right way to think about it? That for the brain to learn how to see?
I think that's a good way of looking at it. In a sense, the brain, what actually happens is the brain learns to see through only one eye
Yeah.
because the inputs from the two eyes are no longer in proper correspondence. So it's still learning, and it's learning to essentially disregard one of the inputs, but of course there are consequences.
Right. And so this happens because of misalignment of the eyes, some sort of asymmetric refraction, things like that, right? So the design of the eyes themselves, something went wrong.
So the sort of definition of amblyopia is a visual impairment that is not apparent in the eye per se. So in other words, the eye is actually fine. The retina is fine. It generates signals that are going into the rest of the brain just fine. So the defect in amblyopia is actually a wiring defect within the brain, and in particular in the visual cortex. Now, the reason that wiring defect occurs, as I said, is because there is not a proper correspondence in the patterns of activity arising in the two eyes. So normally, of course, the two eyes are viewing the same thing at the same time, and you have well correlated patterns of activity coming in through the eyes. But in the case of a condition called strabismus, where the eyes aren't properly aligned, and that causes double vision, so the eyes are not looking at the same thing at the same time, and so there's an imbalance there. And similarly, in a case where there's a refractive error in one eye that by itself is trivial to correct with a lens, for example, a contact lens. But if it goes undetected, you have this situation where only one eye can form a crisp image of the world and the other eye is fuzzy.
Mm.
And the consequence is, again, this miswiring that occurs in the brain. And probably the most severe cause of amblyopia is a unilateral cataract. So there's a clouding of the lens in one eye. And so it's just impossible to form an image on that eye, even though, again, the eye itself, other than the lens, the eye is fine. Again, it's not difficult to correct a cloudy lens these days.
Mm.
But if it's not detected or corrected early in life, then you'll have a permanent visual disability.
Right. So I don't know a lot about this, Mark, but oftentimes one of your eyes is more dominant than the other, right? I remember when I had glasses for the first time, my right eye was much more powerful compared to my left eye. So when there is some sort of a asymmetry like that, would that also cause some level of loss, in terms of learning?
Yeah, I think that that would be an example of the imbalance in refraction in the two eyes.
Mm.
So, for example, I also have refractive error, and I wear one contact lens to correct that, so it balances the two eyes. So my vision's fine. But had that refractive error occurred during infancy
Yeah.
and gone undetected, there would be a good chance I would develop amblyopia.
Mm. So that is the issue. And so if you don't pick it up, so if you had the issue in infancy and early childhood and you don't correct it then it's much more difficult later, right? That's the issue that the wiring, sort of gets less plastic.
That's exactly right. That's exactly right.
And so how do we deal with it now? So first of all, what's the incidence rates of this, Mark, generally speaking?
Oh, approximately 3% of the population is develops amblyopia.
3%. So that's quite high. And so these are adult population, and we have a condition. They have this condition. So what's the corrective measures, or is there any sort of therapeutic intervention for it?
Right. So um, if it's detected, if the cause of amblyopia is detected early, it can be corrected. So, for example, as we were using the example of a cataract, for instance,
Yeah.
obviously a cataract's pretty obvious, and if you are lucky enough to grow up in an area of the world where there's good medical care, then that cataract can be corrected in an infant, and there'll be no permanent visual disability. So the visual system will develop normally. Similarly, if the strabismus is developed, so the eyes are crossed, for example, surgery can be done to weaken one of the muscles that controls the eyes and restores the alignment of the two eyes. So that can be corrected again in infants. And finally, if there's a refractive error, that can also be detected and corrected with lenses. So if the cause of the imbalance in activity from the two eyes is discovered early enough and corrected early enough, there need not be a development of amblyopia. Unfortunately, sometimes even with early detection and correction, amblyopia can arise. Um, and so then you have to develop, treatment strategies to try to restore vision in the weak eye, the amblyopic eye. And the classic therapeutic approach is to patch the good eye, put an eye patch over the dominant eye,
Mm.
the so-called fellow eye,
Yeah.
and force vision through the amblyopic eye. And this can promote recovery of vision through that eye. So it's not sort of the good news is that the deficit can be reversed, but it only works if that patching is done early in life, before the age of approximately 7 or 8 years of age.
Mm.
Um, After that, there's a very poor prognosis when those connections sort of solidify.
Yeah. So symptomatically, if you have this condition, one of your eyes is functioning, so you have less depth perception and things like that. Is that what happens?
Yes, you have a failure of stereoscopic depth perception. We use cues in the environment to determine depth, and that's fine.
Yeah.
Far objects appear smaller than close objects, but for precise stereopsis requires a good image formation in the two eyes to calculate depth. And so, amblyopia, you are left with one good seeing eye.
Mm.
So it's not total blindness, but there is a lifelong disability associated with it. And of course, one of them is loss of stereoscopic vision, and that can limit career opportunities. It can have negative impacts on academic pursuits. And of course, there is a great risk of total blindness if the otherwise healthy eye is damaged over the lifespan.
So this refraction error you talked about, is astigmatism related to that or no?
It is related in the sense that astigmatism um, is a irregularity in the surface of the eye, so that not all parts of the visual field are focused at the same on the same plane on that sheet of film at the back of your eye, the retina.
Yeah.
So these are all fall in the category of refractive errors. It can either be too much refraction, too much bending of light, not enough bending of light, or an eyeball that's too long, or an eyeball that's too short, or an irregularity in this in the surface of the eye that bends the light. So all these things can contribute to amblyopia.
And is there a significant genetic component to it?
Well, the I would suspect there is a significant genetic component to strabismus, the sort of misalignment of the eyes that comes with different muscle lengths. That could easily have a genetic basis, but the causes of amblyopia are sufficiently diverse that many of them are just sort of errors of early development that are not necessarily clearly tied to genetics.
Any diet or nutrition related things that we know of that might cause it?
No,
Okay.
not to my knowledge.
Yeah. So this is an issue that the brain has wired itself, given a sort of um, I should say non-optimum inputs. They do the best it could. And once the person is beyond 10 years, 12 years old, it's really difficult to reverse it, right? So, are there methods? You talked about surgical weakening of certain aspects, but uh, are there any therapeutic interventions to increase plasticity, or that is not possible?
Well, actually, the brain has a considerable potential for plasticity, even after the age of 10, which we should all be grateful for, since if we remember this conversation, it will be a consequence of that plasticity. Um, so the challenge has been to try to re-engage those mechanisms that may lay dormant. Um, and there's been tremendous progress in this field to achieve that in recent years. And one of the reasons that we study amblyopia, or I should say the consequences of early monocular deprivation, is that first it's a example of the critical role of sensory experience in the final wiring of the nervous system.
Hmm.
So students of nervous system development have been fascinated by this phenomenon for well over half a century at this point. Um, and as I say, it's not necessarily basically the brain is wiring depending on the information that it has available to it. And if you have a defect in the sensory organ, that's going to degrade that information, and it's going to essentially take advantage of the input that it has to achieve optimal vision, even though one eye gets left behind. So it's not an example of abnormal development. Really, it's a example of normal development under abnormal circumstances. So it's, as I say, it's been of interest to neuroscientists that are trying to understand the role of experience and neural activity and refining connections in the brain. And also because it's such a dramatic example of the impact of altered sensory experience, it's been studied in the context of learning and memory for a long time as well, with the assumption that if we understood better how experience and deprivation make synapses stronger and weaker, we would gain tremendous insights into how synapses are modified in the brain during the memory formation, for example. So it's been a very rich area of study. And as you say, one of the features of it, prominent feature of it, is that this amblyopia, or this visual impairment, is a consequence of altered visual experience during early development. So if the same alterations occur in adult, for example, I gave you the example of my eye where I wear one contact lens, this developed as a consequence of aging. And I'm not going to become amblyopic as a consequence because my brain, those circuits are no longer sensitive to that abnormal visual experience. However, the sort of flip side of that, I mean, on the one hand, that's great. We're not vulnerable, as vulnerable in adults as we are to children in abnormal sensory experience. The flip side, unfortunately, is that the prospects of reversing a deficit diminish as we get older. And your question really is what's being done to try to promote recovery of juvenile plasticity to allow for recovery of function. And it's a deeper question than just amblyopia, because it applies to recovery of function after all sorts of brain diseases, damage, injury to the brain. So again, it's a system that we have to study processes that are extremely important from a neurotherapeutic point of view. So in recent years, there's been a lot of progress in understanding the factors that bring to a close this period of sensitivity to abnormal visual experiences. We call it the critical period. So as we said, in humans, it ends at about age 8. So there's been a lot of progress in identifying the factors that start to constrain the degree and type of plasticity that's permissible in the brain after that age. And with that knowledge has come some sort of exciting avenues of discovery of ways to sort of reopen or take those brakes on plasticity away and allow potentially plasticity to take over to restore vision in the context of amblyopia, or brain function generally in the context of brain injury.
Right. Yeah. So like you say, it's a broader question. It's about sort of a systemic aspect of the brain. It seems to be really good at learning things early on. It gets sort of locked in, and then once it's locked in, changing things are much, much more difficult. And we see that in all aspects of the brain function, right? So you another interest you have is the synaptic substrates of visual recognition memory. You say detection of novel stimuli that may predict reward or punishment requires long-term memory for and recognition of stimuli that are familiar. So talk a little bit about that. So detection and familiarity recognition, you say, are often impaired in neuropsychiatric diseases. So understanding those things are quite important. So you're going through life, sort of running experiments, Mark. When I think about it, some of those experiments succeed, some of them don't. So there is a positive and negative reinforcement happening all the time. And if that is not happening in some systematic way, that could lead to issues.
Yes, I mean, let me reorient this a little
Yeah.
bit, and I because I think this is an interesting illustration um, of the progression of science, actually. Um, and so when I began my career, many decades ago now, the burning question was to elucidate the mechanisms by which visual deprivation and experience modify connections in the visual part of the brain.
Mm.
And that was my singular focus as a scientist,
Yeah.
was that question. And but of course, over the years, you make along the way unexpected discoveries. They derive from the core interest, but it gives you an opportunity to form a branch off the main trunk of the tree.
Mm.
And some branches you choose to ignore. Some branches your students will pursue. And then some branches you say, God, this is too interesting. I can't, I can't resist.
Right.
So, um, so these experiments on visual recognition memory fall in that category. So, um, when some years ago, the field that was studying the synaptic basis of amblyopia um, really converged on the use of mice as an animal model.
Mm.
They have a number of advantages for doing this kind of mechanistic study. Um, I will say they have the disadvantage of not being particularly visual species. So
Mm.
they have actually a pretty lousy visual system, but we use it anyway. And um, along the way, a student of mine, who is now a very successful professor at Columbia named Nate Sawtelle, made a really unexpected discovery, which was he would record the responses in the visual cortex of the mouse to a very simple stimulus, which is just a pattern of stripes. We
Mm.
call it an oriented grading stimulus. And he would record the activity elicited by that stimulus. This is an awake mouse, and then mouse was put away, came back the next day, and he would record again. And we were not expecting that response to change.
Mm.
Um, but what he discovered was that every day he came back, the response would get a little bit larger and a little bit larger, a little bit larger,
Right.
until it reached an asymptote.
Mm.
And we, there were a lot of reasons that could have been an artifact, but um, Nate and another graduate student at the time, Misha Frankel, had the good sense of saying, I wonder if what would happen if we shifted the orientation of that grading to a novel orientation.
Mm.
And what they discovered is that if you shifted the orientation, even by just a few degrees, the response was back at what was the same as the initial baseline. So he discovered this really, really dramatic form of experience-dependent plasticity in the visual cortex.
Mm.
So this response was really growing. I mean, it could almost double in size, which is not a subtle effect.
Mm.
And so we called that stimulus selective response potentiation. And we have, I think I can't remember when it was discovered, well over 10 years ago. And we've been studying it ever since. And one of the, I think in a sense, it was a phenomenon that was for which we didn't really understand the significance
Mm.
until more recently.
Mm.
And thanks to the work of others, in particular, Sam Cook, who's a professor at King's College in London now, We found that if animals are given the opportunity to explore these stimuli, the first time they see a stimulus like that, they're quite interested in it and they'll run up and explore it. But with successive days, they lose interest because the stimulus itself conveys no relevant information. It's not rewarded, it's not punished. Its novelty is worn off.
Right.
And so what we are observing is the changes in the brain that occur when an initially novel stimulus becomes familiar. And familiarity, recognition of familiar stimuli that confer neither reward nor punishment is extremely important so that we don't devote computational resources to those stimuli.
Right.
We really are tuned up to detect novel stimuli, and that's what this process is actually used for. So it's used to filter out meaningless information and be primed to attend to novel stimuli. So we call it visual recognition memory. It's also, you could call it mechanisms for novelty detection. So basically two sides of the same coin. And this is a very fundamental process that we depend on, and that can go awry in disease states. So you mentioned schizophrenia,
Yeah.
autism is another example, where there's a difficulty in sort of coping with novelty and learning to habituate, learning to no longer react to stimuli that are non-threatening.
Wow. Yeah. That is really fascinating, Mark. So, the problem to solve, even in the computer science world, is really discarding information efficiently, right? Uh,
Right.
so the algorithms that are being sought is really efficient discarding of information, because information overflow is the biggest problem from a decision perspective. It's the same problem for the brain, right? So a very efficient brain would, like you say, would recognize familiarity very quickly, and don't really devote computing resources to those stimuli, so that they can optimize around novel, if I understand this correctly, Mark, novel stimuli, and that's useful in the variety of ways, I would imagine.
Yes, that's absolutely correct. And we just feel so fortunate that we have an opportunity to study this process. You know, it's a fairly low level decision of recognizing novel stimuli, but it's also fundamentally important. And we just have a very robust way of studying the mechanism now.
Yeah, so the neuropsychiatric diseases, schizophrenia, autism, you mentioned, these are situations where the brain is unable to do that. It's basically reacting to stimuli it has seen before, sort of the same fashion as it would to a novel stimuli.
Yes, I mean that actually is a feature that was recognized very early on in schizophrenia. Is essentially a difficulty in essentially modulating a behavioral response to familiar innocuous stimuli.
So does this lead us to some sort of intervention for those diseases in any way?
Well, we that's the hope. We're we're not there yet. I think that we have learned enough about this process in the mouse visual cortex to recognize that it can be disrupted in a lot of different ways. So there's a lot of ways, not surprisingly, there are a lot of ways to break a machine. And I think the challenge that we have is understanding, which of those potential defects are the cause of pathology in a particular disease state. And so something, in other words, someone that is given a diagnosis, say of schizophrenia or autism, um, has a broken machine, but the cause of that disruption can vary from one individual to the next. And so the appropriate treatment will vary from one individual to the next. And I think the challenge that we face in the field is trying to parse the patients that have these disorders into groups that we could predict would respond to particular therapeutic approaches.
If information overload is one of the issues here, Mark, I wondered, could there be some sort of external filters? In other words, if the information going into the brain is mediated in some way, would that have some effect?
I think that's an interesting suggestion.
Yeah. I don't know anything about it. I'm just asking questions.
Well, I don't want to. I don't want to get too far away from what I'm competent to
Yeah.
discuss, but you can imagine that sort of limiting sensory stimuli could have a beneficial effect for people that are unable to process a particularly rich sensory environment.
Yeah.
I think, for example, in the case of at least some cases of autism, where there can really a big problem is sensory overload, and obviously the strategy that's adopted by all caregivers of people that are affected that way is to try to limit sensory overload.
Right.
Like you don't take your kid to Disney World. So that's essentially that's being done empirically. But what we'd like to really understand better is what's going on in the brain, and are there some clever ways that we can restore normal function.
Yeah. Yeah. I'm just thinking, as artificial intelligence progresses, you could have a companion, let's call it a machine, that is able to sort of learn where the optimum inputs might be. And so, you could have a companion machine that produces the right amount of input into the brain, and it could learn from the feedback it's receiving as well. It could be interesting in the future to think about things like that potentially.
I think that's a very interesting suggestion. I'm thinking in terms of what comes to mind is the sort of sensory overload that would come with a pilot, for
Yeah.
instance, particularly in a combat situation where there's a lot of incoming signals and trying to filter, what's a flock of seagulls and what's a missile that could kill me,
Mm.
and then modulating the alert that is available to the pilot to make a decision.
Right. Right. Yeah, it's a fascinating area. We'll take a quick break, Mark. When we come back, we'll talk more about autism.
That sounds great.
Thank you. This is a Scientific Sense podcast providing unscripted conversations with leading academics and researchers on a variety of topics. If you'd like to sponsor this podcast, please reach out to info@scientificsense.com. So we are back. Mark, we were talking about the brain, its ability to use information, and as it uses it, it gets locked in, it loses its plasticity, and that has implications for diseases like amblyopia, that happens in infancy and early childhood. But as people grow up, it becomes a more difficult thing to intervene and correct. And similarly, there are certain neuropsychiatric diseases like schizophrenia and autism, where the brain appears to be sort of overstimulated, because it couldn't really discard familiar information or familiar stimuli that it has already seen. So it has to deal with a very large amount of information, and that might be sort of an underlying condition for some of those diseases. The area of autism, it's now largely called autism spectrum disorders, ASDs, and intellectual disabilities, IDs. Um, is another area of your interest, and your lab is doing a lot of work in this area. Um, you say one of the main barrier has been identifying the defective cellular processes within the brain that disrupt behavior and cognition. Increasing evidence indicates that many cases of ASD and ID have a genetic etiology. So, you want to talk a bit about what we know about, kind of the direct connection between the brain's design, brain's function, and these diseases?
Sure. So we, I sort of gave the example before of this path of discovery where we began studying visual deprivation. We had one branch of our tree that came off to study visual recognition memory, and another branch of the tree that came off was the study actually of a specific genetic cause of autism and intellectual disability called Fragile X syndrome. And so a word about Fragile X. A Fragile X is a monogenic disorder. So it's a disease of brain development that is a consequence of the silencing of a single gene called FMR1 that encodes a protein called FMRP.
Mm.
And we were studying the mechanisms of experience-dependent synaptic modification in the context of our studies of visual development. And one of the phenomena that we were particularly interested in is it's called long-term depression. It specifically refers to a activity-induced decrease in the strength of synaptic transmission.
Yeah.
So weakening synaptic connections. And we were pursuing a mechanism for a form of this type of LTD, we call it. And we made a number of discoveries at the time that we didn't anticipate. And one was that this particular form of plasticity required the immediate synthesis of new proteins at the synapse.
Mm.
And so we became interested in what are the mechanisms by which activity can regulate the synthesis of proteins at synapses. So, purely a basic research question here. And, another one of the, wonderful rewards of a scientific career where, now we had to learn about protein synthesis regulation. So in our research, our search of the literature, we came upon this protein, FMRP, as being a protein that is, in a healthy brain, normally synthesized in response to activity at the synapse.
Mm.
And so we thought, well, this is an interesting candidate for a critical protein in this form of plasticity.
Mm.
So another example of how science happens, I happened to give a research seminar at a meeting about some of our work, and unbeknownst to me, I was sitting next to the geneticist who discovered the mutation responsible for Fragile X, whose name is Steve Warren at Emory University.
Mm.
And Steve, leaned over and said, well, that was a really interesting talk. Would you like to look at the Fragile X knockout mouse? So a mouse that was the model of the human disease.
Yeah.
And I said, well, yeah, that sounds like a great idea. So we started studying these Fragile X mice, and the trail gets a little windy here, but basically the bottom line is that in the Fragile X mice, there appeared to be too much protein synthesis at the synapse. So, in other words, the FMRP protein normally serves as a brake on protein synthesis. In the absence of that FMRP protein in the disease, it's essentially like losing the brakes on a car. So you tap the accelerator and the car goes careening down the hill out of control with no brakes. And that's the analogy of what was going on at the synapse.
Right.
So that was unexpected, but we knew something about what the signals were that regulated that protein synthesis. And we had the idea that perhaps if we inhibited that signaling in the brain, we might be able to correct not all of Fragile X, but many features of Fragile X that we could ascribe to altered protein synthesis regulation. So this became known as what's called the mGlu-R theory of Fragile X. mGlu-R is a abbreviation for metabotropic glutamate receptor, which is the key molecule. So we set out to test this idea. We and many other laboratories around the world set out to test this idea, and we're surprisingly successful. So we found that many deficits at many levels of analysis in Fragile X animal models could be corrected by inhibiting this neurotransmitter receptor. So we were, of course, buoyed by that success, and we started wondering if defective protein synthesis regulation might be a common theme in other genetic causes of intellectual disability and autism. And so we started digging through the literature about what was known at that time anyway. And indeed, a number of other monogenetic disorders that are characterized by intellectual disability and autism, such as tuberous sclerosis complex, Rett syndrome. These are disorders that also could be sort of easily connected with the idea that a fundamental problem is altered protein synthesis regulation at synapses. So we were very excited about the possibility that maybe therapeutics that we could discover by studying Fragile X might be more broadly useful for other causes of autism. Now, of course, we now know autism is extremely complicated. There are hundreds of different genes that are implicated. Not all of these will converge on altered protein synthesis regulation, but many do. And so it's kind of like the example I was giving you earlier about there's more than one way to break a machine. But in the case of autism, for example, maybe there's 6 different ways that we can disrupt circuitry that ultimately manifests as autism. Well, then if we could understand what those 6 things are, which of them would respond to a particular therapeutic approach, we could make some great progress there. So that's a question that fuels a lot of our work.
Yeah, it's like you say, the machine can be broken in many ways. It's a machine that's finely tuned, finely positioned on a knife edge, can go in either direction very quickly. I was just thinking, Mark, that Alzheimer's disease research in a direction that attempted to do things, at least without knowing a lot about it, in a similar vein to reduce production of amyloid plaque or other things in the brain. It hasn't really yielded much for the disease, right? Do you see this to be different?
Well, I should say hope springs eternal. I will say this: we had really exhilarating results in the animal models of Fragile X. And we have a lot of advantages in Fragile X because, in the sense that we know the cause of the disease. We have good animal models of the disease because it's a single gene disorder. And many of these approaches that we were sort of pioneering in mouse models of the disease also worked in other animal models, including a fruit fly model of Fragile X and a rat model of Fragile X. So we we had everything going for us in terms of sort of reducing the risk in translating the discoveries from animals to humans.
Yeah.
So clinical trials were done using these same inhibitors, and they were not successful in Fragile X.
Mm.
And this, of course, was a big disappointment. But it's important, in my view, it's important that we don't throw the baby out with the bathwater. You know, there's a number of people throw up their hands and say, oh, another example of a failure to translate a mouse discovery to a human. But I think it's very important to keep in mind how little we know when we cross that bridge into doing a human study. For example, I mean, some trivial questions like, what's the right dose of the medication to use?
Mm.
What's the right duration of treatment before you could expect to see a benefit? We're talking about brain development here. Um, you know, are there side effects that will limit the exposure of the brain to this drug? And very importantly, what are the right metrics to measure an improvement in
Mm.
function? And so these were all unknowns when these studies began. And now we know a lot more. So actually, I think we're very well positioned to have a success in Fragile X. So I'm personally quite optimistic that we're on the threshold of a success. And I think it'll not only, I mean, of course, it'll be wonderful for individuals with Fragile X and their families, but it will also have the effect of essentially validating the usefulness of the animal models in this disease.
Yeah, and I would imagine, Mark, that the timing is also important, right? So the earlier, the better from an intervention perspective.
Definitely, definitely.
And so, yeah, so, I don't know what clinical trials, what the characteristics of the population there was. Were those in certain age bracket, or what was the characteristic?
Yes, I mean, that's a great question, a great point. Of course, when you're doing a clinical trial, particularly one with a new medicine that's never been used in humans before,
Yeah.
the number one priority is safety. And so, the FDA holds us all to a very high standard to ensure that an experimental medicine is safe and the safety can be demonstrated for the duration of the study.
Yeah.
And they're very reluctant to give you the green light to go into a pediatric population with a new therapeutic entity or
Mm.
modality.
Right.
So the early studies to treat Fragile X were by necessity done in older patients.
Right.
So many of them were adults, middle-aged adults. They could go down as young as late adolescent. But the I think until there was enough safety data,
Mm.
there was no chance to go earlier. Now there are studies underway where they're using much younger subjects with some of these same medicines that failed in the older subjects. I should call them subjects, patients.
Right.
And then there are other molecules that whose safety actually has been established
Mm.
and have been green lighted for younger studies in younger patients. And some of these studies have come actually very, very close to clinical success. So that's what fuels my optimism.
Yeah, yeah, it's really exciting. So in conclusion, Mark, you are doing work in a lot of interrelated areas, all related to foundationally how the brain works, so to speak. How does it process information, and when things go off kilter, so to speak, whether it's able to come back from that deficit in some fashion. So if you look at all this research that you've been doing, Mark, what is the area that you are most excited about, that we will feel, you feel rather, that we'll get some, sort of practical human therapies for the diseases that we discussed, maybe next 5 years, 10 years?
Um, in a way, you're asking me to, choose my favorite child. But since you put a finer point on it, where do I expect to see successes in the next 5 or 10 years? I certainly expect to see a success in Fragile X.
Yeah.
In that time frame, and I also am quite optimistic about some brand new therapeutic approaches in amblyopia. That we didn't get a chance to talk in detail about that, but there are some really exciting results that show that it's possible to get a full recovery of vision in an amblyopic eye at an age that's older than what would be possible with patch therapy, which is the current standard of care. So I'm I'm an optimist by nature, and I'm very optimistic that within the next 10 years, we'll see some therapies advanced on the basis of this research.
Excellent. This has been great, Mark. Thanks so much for spending time with me.
It was really a pleasure, and thank you very much for your interest in our work.
Absolutely. Good luck with this very important research. Thank you. This is a Scientific Sense podcast providing unscripted conversations with leading academics and researchers on a variety of topics. If you'd like to sponsor this podcast, please reach out to info@scientificsense.com.